Dr. Jason Aldred is a movement disorder neurologist from Spokane, and he is the co-owner of Selkirk Neurology and the owner of Inland Northwest Research.
My name is Jason Aldred. I wanted to first thank the APDA for inviting me out here and you all for joining us today. I am from Selkirk Neurology and Inland Northwest Research. It's my private practice and medical research company based in Spokane, Washington. It's Eastern Washington. Is anyone here from east of the Cascades? Couple back there, and hopefully maybe some more online. Okay, excellent. And also, I teach medical students in the second year of movement disorders and Parkinson's disease at the 4-year WWAMI program based in Spokane, and Washington's newest medical school, Washington State University, based in Spokane. So work with both of those schools, talking to those students about Parkinson's and whatnot. Well, today I'm going to talk about therapies that may slow Parkinson's progression, next generation therapies, and we, in theory, could be have been saying that for years. But Dr. Zibatian's talk already today, I think, has shed light on that we're in a very different time now for Parkinson's research. Specifically, his conversation about precision medicine, specialized medicine of sorts, is certainly one of the exciting new angles for Parkinson's research. And there's still a whole need for everything else, the other group, right? Ways getting at patients that don't necessarily have LRRK2 mutations in GBA. So we're trying to do therapies that might work on many different angles, for different patients. I want to talk about two different kind of categories here. One of these, the first one is the immune system and immunotherapy. And so just as a brief introduction, our immune system is really second only in complexity to the human nervous system in the human body. So there's, learning this in medical school, we learn about all the many pathways of the nervous system and all the many pathways that the immune system can go down. And to the point of perfection, in a way, our body uses its immune system to fight off external invaders, virus, bacteria. And also, we don't really recognize this as much in the general public, but our immune system's crucial for controlling cancer and other things within our body that doesn't go properly. And so when we're talking about neurodegenerative disorders and systems within cells in our brain that are supposed to function appropriately, when they do not function appropriately, hopefully there's some kind of gap mechanism to fill that in. And that's where we're going to be focused with immune therapy and trying to actively direct that. So again, immunotherapy has revolutionized our treatment of autoimmune disorders and cancer. We use mechanisms similar to the body's own defenses, so using our own innate tools to specifically target the unwelcome protein, hint, alpha-synuclein, perhaps, or LRRK2 or some other mutant proteins. And then, this is where it's important, right? I mean, to be able to do this is one thing, but to be able to dose it, to really titrate it, to individualize the therapy, to allow for a careful reduction in the amount of abnormal protein to prevent further damage. Because as you've heard already, many of these proteins they're supposed to be there. They're just not behaving appropriately, or because of a variety of reasons, they are formed in ways that can put us at risk for Parkinson's. Alpha-synuclein. Raise your hand if you've heard of alpha-synuclein. Okay, we're getting even more hands up there progressively. So when we're kind of coming to a unified theory or something that unifies Parkinson's, there's truly nothing truly unified in Parkinson's, but many things approach that. And alpha-synuclein is certainly one of those, an abnormal protein that we have normally in our bodies, but abnormal in Parkinson's. In particular, the way that it's kind of clumped. So I'm using the term clumping to just refer to Parkinson's disease forming kind of at-risk structures here, this alpha-synuclein monomers. Monomers here. The alpha-synuclein monomers are normal functional proteins that perhaps help nerves transport things, and we don't exactly know, but function appropriately. However, there are, for certain circumstances, not completely clear, but certainly gene mutations would be one of them, alpha-synuclein oligomers. So they kind of clump up, and then this can be transported outside of cells and put us at risk for developing these fibrils, and those go on to form Lewy bodies. And who has heard of Lewy bodies? I think that beat alpha-synuclein for hands, so we've made it. So Lewy bodies are certainly associated with decreased cell survival, cell death. But the interesting thing here is before even getting to Lewy bodies, we see that these alpha-synuclein oligomers, so these clumps, right? Clumps don't sound good, and they're not good in our cells, but they place our dopamine cells at increased risk for cell death. Just a little technical piece here. Alpha-synuclein is something called the Brack hypothesis. So I was a fellow at OHSU when the Brack hypothesis came out. This is a theory about the spread and progression of Parkinson's. We know that abnormal alpha-synuclein and Lewy bodies are not healthy for us and put us at risk for Parkinson's and are associated with the actual clinical features of Parkinson's. But why do people with Parkinson's progress at different rates? We heard in many of these meetings there's thousands of different types of Parkinson's, one for every kind of person or more. And really there is some truth to that of sorts, but there are factors that influence progression. And so why do some people on one end of the bell curve progress really rapidly, but they still have Parkinson's, and other people progress extremely slowly over decades? And that's due to some factors within our bodies that must vary. And perhaps the progression may relate to the spread of alpha-synuclein oligomers, or these clumps, from one cell to another, killing them. We have premotor symptoms of Parkinson's. So these are symptoms of decreased smell, acting out dreams at night, bowel movement issues, other problems as well, mood symptoms. And these clumping of proteins are found in cells outside of the dopamine system as well. Again, getting to the point of actually changing the progression of Parkinson's is not going to focus solely on the dopaminergic system, the dopamine system. It's going to focus on many different systems. Ultimately, things that really relate to two of the greatest fears of people with Parkinson's is not having mobility issues and serious thinking issues. So those are two of the big ultimate factors of Parkinson's beyond what we can currently treat pretty well that we really have a need to get at. The idea, though, coming back to this alpha-synuclein and Brack hypothesis, is the moving of abnormal protein from one cell to another. This may actually provide a unique opportunity for treatment. This is just a picture of what happens on the top slide here in Parkinson's disease when we see, supposedly within cells, alpha-synuclein here, monomers here, these are okay, then they start to clump and they spread from the affected cell outside of the cell, clump even more, aggregate, clump even bigger into the Lewy body. So using an immune system type model, what we are starting to see interest in and results from many, many years of folks in the lab is these clumps going outside of the cell and then being bound with antibodies. Antibodies, of course, are produced by our own immune cells, but can be synthetically produced as well, targeted directly to something that we want to isolate and harness and tag onto other immune cells in the body to say, hey, take this out, let's remove this before it aggregates and causes toxicity to our dopamine system and the other cells and spreads. So again, a way to slow progression in Parkinson's. Again, the idea here is that there are certain types of glia, there's certain types of immune cells, microglia, that get activated in Parkinson's, and we've heard about immune activation or inflammation. Inflammation is seen in Parkinson's disease. And the thought at some time was maybe that was somehow a trigger, or maybe it's an after effect. Well, when dopaminergic cells and other cells in Parkinson's are affected, there is definitely a response when cells die. So it could be that you might target one specific, the spread of a Lewy body from one neuron to another, but if you don't also dampen down the inflammatory pathways, you could still have other damage to other cells. So in Parkinson's mice, we see that overproduction of this protein, alpha-synuclein, there's increased microglia activation as well. And so this the glia are supposed to digest. They're kind of the garbage can, if you will. They're supposed to digest this abnormal protein, but when it becomes clumped big enough, they no longer can do that. They can no longer achieve their goal of removing this out. So if we can get to these protein clumps before they get too big, for lack of a more precise word, this might really allow us the point to intervene from the spread from one cell to the other outside of the cell. Now, when we start to talk about immunotherapy, we think about other things like vaccines and and the term Parkinson's vaccine has been thrown around. That was the first way that I remember in looking at a paper from Austria where they used an actual active vaccination, looking at ways to target alpha-synuclein. Well, certainly it would be nice to have a one and done type thing, or maybe a vaccine with a booster shot. There are some problems with that, so I just wanted to speak to this in this presentation. There's some variation between immune systems, and particularly, it just so happens that people with weaker immune system are often older, and some people respond differently to vaccines. So there's a lot of variation in response, and in the small studies that have been done so far, one of the concerns is that, like our immune system varies in how we respond to a vaccine, it may vary considerably in the response to like some type of therapy targeted to make the body's own system recognize the alpha-synuclein and remove those. Really what we're talking about now in terms of using an immune therapy is more in line of like something we call passive vaccination. And again, this is more appropriately termed immune therapy, but I wanted to bring this point out because I find patient after patient will kind of bring to me this concept of this vaccination, and I wanted to put it in those terms. So given after exposure to an agent, like a rabies vaccine in this case, so rabies is a virus that can go from cell to cell and cause terrible damage to the nervous system. So after you're exposed to something like that, if you're given a treatment to isolate that and to limit the spread of the disease, that can kind of be this therapeutic window. In Parkinson's, the therapeutic window we obviously want to get to as early as possible. But our hope with any of these therapies that target abnormal proteins is we can get to them at any point in Parkinson's. The later we get there, the less effective they may be. But nonetheless, at any point, certainly earlier is better, but at any point it could potentially be effective. These types of treatments must be given frequently enough to neutralize the damaging substance. So there is some frequency issue here. So as opposed to like a injection, a vaccine type thing you get where you're one and done or you get a booster, the things that are passive vaccinations that don't rely on the body's own immune cells to really generate the initial antibody have to be given periodically. So that's kind of a little bit about how they're different. So just very briefly, our clinical trial group in Spokane is remarkably large and efficient for the size of our area. And so we've got a number of clinical trials. I'll talk a little bit more about them later, some of the other ones, but the phase 2 clinical trials currently looking at this type of technique is the Sparc Trial. So we were part of Biogen's phase 1 study, and also the phase 2 Sparc Trial where we're looking at a compound called BIB54 in people with Parkinson's disease. And we're looking at safety and outcomes. We're looking a bit at disease progression as well. You heard Bob talking just a little bit about that. This is the Biogen trial here. There's another trial by Roche called the APDA trial, and this is looking at a different type of antibody that binds to the different end. So there's different ways to grab something, you know? So the Biogen antibody is grabbing the alpha-synuclein from one end, and the APDA trial from Roche is grabbing it from the other end. So this is a really neat idea. Don't just try one thing only one way, but try a certain therapy like immune therapy from different angles to try to see which one is the best effect. In general, these studies are relatively similar in that they're monthly infusions of these monoclonal antibodies with measurements for safety and clinical effect compared to placebo for over 1 year, and then another year where everyone is converted over to the active infusion. So this is just a kind of a summary here. It's a little bit redundant, but not only are we here in the degenerative state seeing the death of neurons, but also overactivation of these microglia, the immune type cells in the nervous system. And the hope is that with immunotherapies, we can neutralize with the antibodies neutralize alpha-synuclein before it triggers spread neuronal death to the next neuron, and also immune activation. 3:10 today, I know what I'll be doing hopefully. Go Zags. Tyrosine kinase, this is my last piece here, tyrosine kinase inhibitors in Parkinson's disease. Who has heard of c-Abl? Right, okay, we're back down again. So c-Abl is a common protein in any parts of cells with a wide range of function. This is something until, a couple years ago, I last read about in medical school cancer type courses and whatnot and cell biology courses. It's effective, or it's present all over the body. It's kind of a regulator type protein. Proteins do work in cells. In neurons, it involves forming synapses and growth and development, right? So that's something that as children we would certainly need. We've all seen that in disease states, particularly cancer, abnormal overactivity has long been implicated in leukemia. So again, back to the cancer piece, that's been a very informative other branch of medicine, it seems, when we look at neurodegeneration nowadays. Mouse models of Parkinson's disease show promise for neuroprotection looking at tyrosine kinase inhibitors, this type of leukemia drug. However, it's never yet been demonstrated effective in humans with Parkinson's. This is a busy slide. I just wanted to walk you through some big points here. So this talks about sporadic PDA. Again, this is that other group, presumably, if we assume that the GBA and the LRRK2 mutations are responsible for the hereditary forms of Parkinson's. That the sporadic Parkinson's, there is some type of mitochondrial dysfunction. This is the energy-producing part of the cell. There's stress, oxidative stress. We've all heard about oxidative stress for years and years in Parkinson's. It's kind of the generic kind of term for things that damage cells. And what we see that there's increased activated c-Abl protein. And there's a whole lot of bad stuff here that goes on in relation to that, of course. It's trying to handle this oxidative stress. So these are presumably things trying to adapt. Parkin, who's heard of Parkin? Part 2? And so one of the hereditary forms of Parkinson's, this actually is part of this pathway of this mitochondrial overreactive kind of pathway here. And there's other inflammatory markers, the microglia, the immune system of the nervous system, the immune cell of the nervous system is activated. And ultimately, cells may die. Progression with Lewy bodies, alpha-synuclein is also implicated. So what is important about this is notice that this is central, at least in this diagram. It's more complex than this, but this is central. So when we're looking for things to slow progression of Parkinson's or get to the root, perhaps cause, or get to one of the root influencers of Parkinson's, we like things that are central, right, down to the core more so. Now we're going to talk briefly about cancer. And this is where, this is so exciting because whenever I see timelines, I always try to think, gosh, where am I on that timeline? Where are my patients on that? Where are we now, right? Because we don't know. We don't know what the future holds. But certainly with the discovery of this Philadelphia chromosome in 1960 began this interest more in the c-Abl pathway as the Philadelphia chromosome was determined to encode this BCR-ABL fusion kinase, 1980. And down the line, they did mouse bone marrow transplantation models of this abnormal protein and developed a drug called imatinib. Now, you're a geneticist in neurodegenerative disorders, as Dr. Zebatian, I think, have been very influential in discovering genes in neurodegeneration and pathways, but this is a cancer pathway. So thinking about kind of determining cause, determining frequency, determining how relevant a specific therapy might be versus another. And then, lo and behold, we have phase 1 clinical trial of imatinib presented. Results showing that it influenced the survival in leukemia. And then we start to see the FDA approval, and however, right after approval, what do you see? First cases of resistance reported. Mutations were identified. So the idea of personalized medicine, there really are thousands and millions of types of personalized medicine. And that's the fascinating thing about this. And while even with even if one trial fails, one therapy partially works, we learn a lot from those to go on to the next phase and the next level. And now you have other drugs, dasatinib and nilotinib. Who's heard of nilotinib? Nilotinib, okay. So there is a lot of hype around this medication, nilotinib. I call it the nilotinib hype begins. There is a report by Fernando Pagan regarding nilotinib effects in Parkinson's disease and dementia with Lewy bodies. Well, any first reports on multiple types of disorders is a bit of a red flag because we're thinking, what is this on? What is it exactly about? And so what was interesting though is this was a non-dopaminergic type therapy targeted at the root cause or a root influencer for Parkinson's disease. And let me walk you quickly through this. This is an open label pilot study to test 2 doses of nilotinib on safety. This is a fraction of the dose that we use in leukemia, a fraction. We're using 800 milligrams or so. We're talking about 150 to 300 milligrams of nilotinib. However, this study was really just geared to look at mainly safety, although it was done in advanced Parkinson's patients and those with Lewy body disease, which is a bit of a risky premise because generally we look at people earlier for safety. But what they did find was, in general, it was tolerated relatively well. The primary outcome for safety and tolerability, it generally met that outcome. There were also what we call exploratory. That means let's see if this helps some other factors of the disease besides just safety in an early trial. And they looked at motor function and cognition. And they did report early on that there were some improvements in both of those areas. 12 patients were in this trial, but they were not compared to a control. And that's very, very important because what happens is when this trial was released, there was lots of interest in nilotinib might slow progression of Parkinson's disease. But it wasn't really compared to the control group. It was just compared to where the group started out at. And so we've heard already about Dr. Mishley about placebo and how misleading that can be. So we have to take all of this with a grain of salt. But it was interesting to certainly see that these patients didn't worsen over a 6-month period of time. And also to see that there were some interesting improvements. The mental status score kind of improved compared to the baseline at 6 months. And also there were increased markers for dopamine breakdown at both doses, which is kind of interesting. That's not something that's been measured before. So there were pertinent things to take away from this. But in truth, we really, to this day, know very little about nilotinib and Parkinson's disease, other than the basic science so far and a bit of the early safety data. Current trials, we have both of these at our site, nilotinib for Parkinson's disease. so NILO-PD. This is for people with symptomatic Parkinson's disease in one group, and the second group is going to be a longer trial looking at early Parkinson's. And then a brand new trial that's just started from Sun Pharma called Phase 2, and it's Sparc with a C. Our other immunotherapy trial is Sparc with a K. Why this is Sparc with the C is a little bit of a mystery, given the other trial going on. But this is another TIB medication, presumably, that we think may have a perhaps better safety profile than the first medication. So just in summary, immunotherapy and these tyrosine kinase inhibitors represent two new avenues to slow Parkinson's progression. What's exciting about this is we've got a lot of great dopaminergic medicines out there, but we don't have a lot of non-dopamine treatments, and both of these may target more of the critical aspects that cause neurodegeneration. And so far from what's been released, it seems that these may be reasonably safe, but we have to study it at the next level, look for a bit more outcomes, look at larger groups of people for more safety, and we hope that Phase 3 trials will be on the way soon. All right, and so maybe we can take some questions. That's all for my presentation. This is just about our group. We've got a booth out there, Inland Northwest Research.
So I'm curious, if one is positive for a LRRK2 mutation, is that de facto the reason that you have Parkinson's, or is that maybe the reason you have Parkinson's?
So the answer is it's a contributor. It's a big contributor, but as you saw from the slides that I showed, over half of people who have a LRRK2 mutation, at least the G2019S mutation, and live a normal lifespan won't get it. So it's a big contributor, but it's not the only thing. And the same is true with GBA. GBA is a contributor. It's not as powerful a contributor as LRRK2, but it's a big contributor.
I forgot where I read about the results of the study, but it had to do with the tagging of certain cells for destruction. And they found that the alpha-nuclein molecules were often not tagged for destruction, and they figured out a process by which that could be done, which would eliminate a lot of the alpha-nuclein cells. Have you heard about that? Either one?
Yeah, so one thing I can say is that a lot of, when mutations in different genes are found that cause Parkinson's disease, researchers try to tie that back to synuclein. And so probably synuclein plays a role, it interacts with a lot of these different genes. I think the idea that you just heard about was that synuclein is important for normal function at the synapse, for cells to communicate with each other. The idea is that it's good, but if it aggregates into these clumps, that causes problems with the cell. So anything that can cause synuclein to aggregate, to clump more than it should, is a bad thing. But that could happen by many different ways. With GBA, the idea is that it's causing these lysosomes, this waste removal system, to get clogged up so that synuclein is aggregating and not being cleared as fast, but other proteins are too. So that's one of the ways in which synuclein that's essentially tagged for removal doesn't end up getting removed as fast as it should be.
You know, it's an observation from all of us that are not the professionals, is that some Parkinson patients have a huge battle versus others that have a lighter dose. Is it, explain what you were just sharing with us, all these variances that the individual people have that are in the experiments. It's not that one's got a bigger dose of Parkinson's, it's how their body is susceptible to it.
So I could just speak to one aspect of that. Again, geneticists like me tend to explain everything in terms of genetics. So so that's my bias, but let me give you an example. So people with GBA mutations, if you take a group of people with GBA mutations with Parkinson's disease, and you take a group of people with Parkinson's disease who don't have GBA mutations, the people on average with GBA mutations get worse faster. What I mean by that is that their motor symptoms get worse faster, their thinking and memory declines more quickly. So it's absolutely true that there are big differences in how fast people decline. Genetics plays a role in that, but there are lots of other things. Dr. Mishley talked about other aspects, how old they are, what they eat, do they take care of themselves, do they exercise, do they have other diseases, do they have high blood pressure, do they have any number of other things. So each person is different, your genetic background, what other diseases you have, what you've been exposed to your whole life that we can't even measure. So those factors probably play a role. It's just that certain factors we can pinpoint and say, aha, you have this, or you don't, but there's lots and lots that we don't understand.
Yeah, I mean, I guess to continue on that point, there are different types of Parkinson's that we recognize, and there's many of those. I mean, you can lump them into people with tremor and those without tremor. And, those without tremor, we see, have a bit more faster progression. It tends to involve cognition earlier, can more aggressively. And I guess the point, I would certainly wonder is that type of Parkinson's mostly affected by like this GBA pathway, not necessarily the gene, but like the GBA pathway. And therefore, even if it's not a gene, but something that influences that pathway, that's going to be the kind of specific personalized therapy for that person. And so that's to your point about like the dose of Parkinson's to getting more, it's going to influence or whatever the specific cause of the degeneration, or the thing that influences the factors that influence that particular form of degeneration over another type, are really where we hope these new therapies are going. And truly, I would caution the group from taking away from this that any one of these might be better than another. It may be that we might find, 20 years from now, look back on the timeline and say, well, there were actually 4 different things we needed to be going after. We were going after 2 at one time. And that's kind of, I think, where we're at now, and wondering, if one of these might, like a GBA, glucose herbicidease GBA therapy, might influence the rapidly progressing patient more than the person with really slow progression. And maybe that works great, it doesn't touch the other patient because there's a whole other different process going on. 2 more questions?
We have Mike back there.
Yes. Genetic testing. So I'll say that actually compared to what we knew 10 or 20 years ago, 9 or 10% is actually a very large number of people. I mean, in the United States, that's probably 100,000 people. That's a lot of people. It isn't everybody, but I think the idea up to this point has been that when people get genetic testing to find out what they have, up to this point, there wasn't really anything that we would do, that information wouldn't change what we would do for you as a patient. It would affect your relatives and whether they were at higher risk for Parkinson's disease or not. But for you, up to this point, it didn't change what we did. It didn't change what trials you were eligible for or what medicines we gave you. Now, that is starting to change. And now the question is, how do we go from where we're at now to being able to screen large numbers of patients with Parkinson's disease in the clinic and elsewhere? And the answer is, we're simply not prepared. There has to be a shift or a paradigm. When you go to your primary care doc, you get a number of standard tests done every time. They check your glucose, they check your white blood cell count, your red blood cell count. So we are going to start to need to do that eventually in the clinic, and I just don't think we're prepared. There's been talk about this, but I think we need to be able to make genetic testing much friendlier, much easier. Another problem is insurance oftentimes doesn't pay for it, and we need to start moving into that direction where we're going to not test you for everything, but at least test you for certain genetic variants that are really important and do it routinely. You may have been tested, but the point is anybody walking into clinic eventually is going to need to be able to have this option. Okay, so one more question?
Yeah, this is a real simple question. When you talk about early Parkinson's, do you mean early onset or do you mean early age?
So we use the term early in two different ways. One is early onset Parkinson's disease, which simply means how old you were when you started having symptoms. And people will sometimes use a cutoff of 50 years or sometimes 40 years. We use the term early in a different context, which is at the beginning of your disease process when you first start showing symptoms, regardless of whether you got the symptoms at age 30 or age 80. So that's, in our parlance, those are the two times and two instances where we use the term early. One more? One more? I'll tell you what, how are we doing it? We can always, we aren't going anywhere, so if you want to come up afterwards, we're happy to talk. I just want to make sure we stay on target. Are we?
Yeah, I'd like to ask about international research. Are you connected with any overseas?
Yes, so we collaborate with researchers in probably a dozen different countries. Some of them are developing countries. Some of those are countries that have well-developed programs of Parkinson's disease. As a geneticist, one of the things that we're interested in is everything you saw here was based on genetics research that was mostly done in people of European origin. We know from other diseases that the mutations in genes that are going to have a bearing on Parkinson's disease in other populations are probably different and perhaps quite a bit different. So we're interested in looking at African Americans and Latinos. And you all have a brochure, right? Can you turn to the front of it? Anybody recognize who that is? Good. So that's Dr. Ignacio Mata. And he was here at the University of Washington in the VA Puget Sound for over a decade, and he just moved to the Cleveland Clinic. But his career is largely focused on trying to look at the genetics of Parkinson's disease in Latino populations. So that's one of the things we think is really important to do, is to be able to not base everything that you see just on the genetics of people in European-derived populations, because there's other lessons to learn, and we need to get that information in order to be able to treat people from other population groups. It's just lagged behind. So what? No, come up.
Let's take on. Thank you so much, doctors, so much. Can you still hear me? I feel like I was louder last time. Maybe my ears are going numb. How's everybody doing? Great lunch? Great. I know, right? Another round of applause for all these amazing presenters. Just a couple short orders of business, and then we're going to have a little break. We are continuing to run a smidge long, which again I see as a positive. It just means we're having lots of good Q&A, and those who you've heard speak, welcome you to come up and ask a few questions if you'd like. They'll also be lingering around after the event so that you can catch them out there. We'd love for you to stop by our sponsor tables, and there's great information and handouts and Q&A that can happen out there as well.